All right, we're going to get started in just a minute. That was effective. Okay. Um, Welcome back from the break, everybody. This next session that we're going to do will be a 30-minute presentation. The session is titled Funding CMT Research, Insight into the Current Research Funding Environment and Ways to Overcome Challenges. Our speaker today will be Dr. Walter Koroshetz, MD, Director of the NIH National Institutes of Neurological Disorders and Stroke. Thank you very much, Walter.
Thank you very much, and a pleasure to talk to you today. Thanks for inviting me out of DC. So I'm going to talk to you a little bit about some of the things that we're doing at NINDS and maybe some kind of futuristic science that we've seen from the project that we're involved in. Just to start off, the Neurological Institute, which I direct, and Sri Ranganathan is here in the back. Really interested in trying to get treatments for neurological diseases. What we do is we support basic translational and clinical research. We put most of our money into basic research, probably around 65, 70%. And that is basic research on how the nervous system works, which is about 22%, and then that's disease agnostic, and about 40% on mechanisms of disease. And I think we feel that that is the best kind of investment to make, because if we don't do that kind of work, no one else is probably going to be investing in that, at least in the US. And that gives us the kind of knowledge base that I think can bring, the things to therapies and taken up by biotechs and then the bigger industries. So our pyramid looks kind of, going up and less money as you go up to translational and clinical, and that's kind of complementary to what the industry has, which little on the basic side, more in the translational, and a lot of money has to go into clinical trials. So that's how we kind of see ourselves. And I guess the other thing to mention that we're really interested in trying to train the next generation, because the tools that we see coming are going to be available for the next generation, and the tools are much more powerful than anything that we have now, or certainly that we had even 5 or 10 years ago. So that's really important, and that is actually one of our bottlenecks, and the thing that we're worried about most in terms of a downtrend in certainly what's happening now in terms of science and NIH. So this is what I talked about, we try not to have any kind of gaps. So in trying to move to treatments, heavily weighted toward the division of neuroscience, which is where the basic research comes out of. But we also have what's unique for NIH is a translational program, which is staffed by people who came in from the pharmaceutical companies. And so these few folks are very adept in the process of moving therapies through to the IND stage. And we have programs to do that, very similar to what was described in Aaron's case, where a lot of work is done by CROs. In this mechanism that we have, the investigator can come in and get access to those kind of CROs. So some money goes to your lab, but most of the money for these projects actually you use to work with the CROs to go through all the stages that would need to get you to an IND. These are milestone programs, so you set it up. This is the kind of things you have to do to get to an IND stage. If you fail one of them, you're not going to get to the IND, and we take your money away. So it is a little high risk to get into this space, but that's clearly the case with therapy development in general. I mean, you just can't make any skips. You have to kind of go through that process. But these projects, to go all the way through the IND stage might run $20 million from us. And oftentimes, as you move along, your program gets more and more attractive to the companies. And our job is not to be a drug company, but to de-risk projects so that the biopharmaceutical groups will take it up. We also have a division of clinical research that does trials. And the area that's probably most relevant here is we have a network that's been set up across the country to do trials in neurological diseases using first in human type therapies. So biotech companies or some academics will come into Neuronext, it's called. They write the grant, they get the grant. Neuronext will do the trial for you with your help and maybe adding sites onto the Neuronext sites, which are existent across the country. And they can do studies in kids, adults. They can do peripheral nerve trouble, you know, um, uh, CNS issues. They're pretty flexible and they've been quite efficient. Okay. Now, there's a lot of changes that happening at NIH. Probably people have read about it or experienced it. So I would say that a lot of changes have occurred and we're not really at a set point, a homeostatic set point now. So I think people have to bear with us as we kind of move into this new set point. A lot of changes have occurred, like they shut down all the reviews we used to do in our institutes. Now they have to all be done in the central services, central review branch. And we have a hard time now getting RFAs out because they have to go through a process and get approved by political appointees before we can make them public. Um, and I don't think anything is going to stop. It's just going to take a long time. There is, unfortunately, also a mandate that we reduce the number of RFAs by half. They thought we had way too many RFAs out. So we have lots of projects, particularly in this translational program, that are very specific, and we're trying to figure out how to bundle them together to reduce the number of RFAs. But on the other hand, I think we can get through all this. It's just going to take us some time. And the good news is that we were able to get 100% of our funds out this year, which our year ends October 1st. So our people worked really hard, got all the money out the door, all the projects. And that's really the most important thing is to get the money out. Yeah. Somebody's happy. Everybody should be happy. Okay. All right. So now I'm going to switch gears and talk about the kind of things we do in the rare diseases. And so the topic I chose was embracing complexity. Probably in this audience, you probably all had to deal with that. 130 different genetic causes of CMT. So that means you're dealing with a very complex environment. And we here at NINDS have probably all your 130 genetic forms in our portfolio. And then we have all these other diseases. So we basically have 400 to 500, 600 rare diseases, depending on how you cut them up in the NINDS portfolio. And so we are pretty active in this space. And I'll just go through some of these things, but these are the things I'm going to be talking about: the Undiagnosed Disease Network, the Rare Disease Consortia, the Rare Disease Clinical Trial Readiness Program, the Bespoke Gene Therapy Consortium, and the Somatic Cell Gene Editing Program. And then the Urgent Network, which is kind of our Neuronext program for gene therapy. So Undiagnosed Disease Network was started probably 15 years ago at NIH. It went through a period of time. It was supported by the Common Fund, and then that expired and it came to NINDS, and we run it with help from multiple different agencies. And this is a program around the country where people who have disorders that defy diagnosis can send in their medical records, they get reviewed, and they get passed out to one of the sites. If they think that they can help that person, often a kid, then they bring them in and try and work them up. And it's been heavily focused on genetics, and they've discovered a whole bunch of new genetic disorders and work up a lot of variants of unknown significance in their model organism centers where they can send things down to a Drosophila or worm model and try and see if the variant that they are worried about is pathogenic in that space. So that's been, I think, pretty pretty interesting, come up with a whole bunch of different disorders. I don't know myself how many were in the peripheral nerve space, but I imagine there were quite a few. 80% of the cases who come in through the undiagnosed disease network over the last 12 years have had some neurologic abnormality. So it's heavily on the neuro side, but it can include other organ systems. Then we work with the Rare Disease Consortium, and that is the consortium itself is run out of NCATS, the National Center for Advancement of Translational Sciences, but they develop the infrastructure. We actually help fund that. And then the grants come in, and then NCATS farms the grants out to the institutes, and we pay for probably half of the grants in the Rare Disease Consortia. And the idea there is to actually do the kind of work that's needed, kind of early stage, to trace the disorders, try and get a sense of the natural history, and potentially work on some early biomarker type stuff. I think it's been, I think, very helpful to the multiple rare diseases that we work with. The truth of the matter is that we have so many that if everybody needed to do a natural history study, we could use all our money for natural history studies. And so that's kind of a problem for us, and I'm sure for everybody else too. But this network, I think, is helpful there. As you know, you have to have, I think it's at least 3 disorders come in together, relevant disorders come in together into the consortium. So this is just going to, the next 2 slides show you what they are. But as you know, we've been funding the Inherited Neuropathy Consortium, which is heavily Charcot-Marie-Tooth, for quite a while. And then you can see what the other ones are with the stars that are related to neurologic conditions. And then here on the other slide, you can see some more. And this program is a little bit similar, but is more focused. This one is called the Clinical Trial Readiness Program, is more focused on getting things in order to package so you can do a clinical trial. So the idea is that if you want, and you guys know this better than anybody else, if you want to go in with a company to test something, you got to have a good package for, how you're going to know if whatever you're testing works or not. And so this is for rare diseases that we think will have a candidate therapeutics ready to test in the next 5 years or so. This is the work you would do. I think they're 5-year grants to try and get yourselves ready so you can go, in with the company and actually get into a clinical trial. Again, this is another one of those, there's a whole bunch of projects that are going to need another quote unquote RFA, and we're trying to figure that out. I would say that that what they're trying to do at NIH as we get rid of the RFAs is to try and put out on a website what they call HTs, which are highlighted topics. So instead of an RFA with all its particulars, you might see a highlighted topic which says the institute is interested in X, Y, and Z, and that might take the place of these previously the NOFOs. So you got to keep your eyes out for that. HTs coming. Then I think there's 5 of them that are out. None of them from us yet. And I think I think we're allowed to put in 5, and we put them in July. We haven't gotten any out yet. But that'll be another place to look for things that would otherwise have been RFAs or NOFOs. Okay. And then in this space, we funded the clinical trial readiness program in CMT. I mentioned NeuroNext, which is a network to test first in human therapeutics. We are attaching onto that is what's called Urgent, the ultra rare gene-based therapy network. And this will fund not just the clinical trial. The clinical trial is the last thing, but it will fund a number of steps that you go through to get the IND from the FDA. So it's kind of like what I talked about before with regard to drugs in our therapy network. This is for gene therapies. So again, you get access to these contract resources. which is probably more valuable than the actual money that you get into your lab. And again, these are milestone programs. And the hope is that as you get through, these are the kind of things that can go into Neuronext and be tested in Neuronext. And so what we did in Neuronext is set up kind of like a consulting group who's pretty experienced in gene therapy. I mean, no one's that experienced right now, but these are good people. And we will then incorporate, if you have a project, an urgent project, into that kind of consulting consortium. Because I guess maybe I'm just paranoid, but what worries me is that there's going to be really bad things that happen, as have happened in the past with genomic therapies, and that could shut down things, and then we really a lot of time will be lost. So I think we need to learn from each other as we do this, because we're really getting into new territory that no one's been there before, and bad things are happening. And so I think that's part and parcel of doing things that are risky for people who have really terrible troubles from their disorders. But I don't think that you can justify doing it on your own right now, because there's so many things that people are learning as they go that you may need to know about, and you want to learn it before something bad happens. Okay, so these are the projects that we have now in urgent. So they're gene therapies for spartoglucosineminuria. We have an ASO therapy for Menkes. I'm sorry, no, that's a gene therapy for Menkes. ASO for prion lowering in Creutzfeldt-Jakob or familial prion disease. We have a program at Columbia to work with Ionis to develop a whole bunch of ASOs for rare forms of ALS, actually 11 different forms of ALS. And then we have a program in DMD patients, microdystrophin, microutrophin. I'm going to jump now to the Bespoke Gene Therapy Consortium. This is a Accelerated Medicine Partnership Program at NIH. Those we call AMPs are consortium of industry, academia, and nonprofits. And the Bespoke Gene Therapy one is really trying to develop a platform for AAV gene therapies. And Terry knows a lot about this group. He's been part of the group, very helpful to the group. But it's a difficult group, right, Terry? But they're working hard to try and develop a playbook for how you get AAV gene therapies to the clinic. What are the things you have to go through? What are the standards? What are the quality metrics? And this is companies working with academic folks. And the hope would be, if this worked, that you would have a platform that you could plug in your gene into an AAV that's been well characterized and know how it's manufactured, and then you can move very quickly to patients. So, and this was really, really popular with the FDA and Peter Marks, and who was a real help in trying to build this. So they are working. You can see all the companies that are interested actually in this space. The AMP programs, the companies put money in. And then these are the projects. Terry's project is on the top because Terry's Terry. It's the one that's furthest along. When we have multiple sulfatase deficiency, propionic aciduria, morquios, and then some of the other disorders in the eye. And that one, so that one is kind of a closed shop right now. They're trying to work the system through, but the playbook they put together may be very helpful to anybody trying to do AV stuff. Okay, now I'm going to jump to what I think is the most exciting thing I've seen, and that's the gene editing program. And so this is a Common Fund project, so it's funded out of the central NIH. I co-chair it with Joni Rudder from the NCATS program. And the idea was to figure out how to translate genome editing technologies into therapies. I think we're in probably 7th or 8th year now. First phase focused on basically technology development, optimizing the editors, delivery methods, developing safety tools, basically off-target effect searches. And the phase two kind of does a little bit more of that, but it's also trying to get things into the clinic, which when I initially got involved, I thought maybe we were going to be out ahead of our skis on this one, but we'll see. So the phase one, so there was work developing large and small animal reporters and testing centers, pigs, non-human primates, for instance, as well as a lot of rodent work, cell and tissue platforms to try and detect off-target effects of different gene editing tools, and a big push on developing delivery systems, which I'll talk about a little bit later, and then expanding the number of genome editors that could be used in the clinic. So that was phase one, which ran for about 5 years. And a lot of really amazing things happened in the technology and gene editing. So that's probably why I'm more excited about it than anything else, because it's changing so rapidly. Like there's one technology, and then 2 months later, something else blows it right out of the water. So the base editing, David Liu, I don't know if people know him, but he's at MIT. His group, they're actually like magicians. They're just coming up with amazing tools every couple of months. So there was first base editing, but then being able to cut and paste, prime editing, and then Steve Ecker with mitochondrial editing, and I forget who it was, but we're actually testing it as an epigenetic editing. And so all these new tools have all come within the last five, well, except for the first one, within the last 5 years. So in the phase two, we're doing technology and assay development, but the real push is on trying to get IND enabling studies, developing clinical trials. And so these are the programs that we're funding. So urea cycle disorders, we'll hear about this. This is out of UPenn, phenylketonuria, and Kat Lutz is looking at a bunch of different repeat disorders out of Jackson each group has oftentimes people from multiple different places. Dr. Yang at Yale is trying to do editing for Angelman syndrome, really interesting project. We have another project on prion disease, and this is an epigenetic editing using the CHARM system of editing the epigenetics, so you can basically turn on or turn off pretty much any protein you want if this all really works. Then we have a bunch of programs in the eye and the ear. And then Jennifer Doudna, who got the Nobel for CRISPR, is actually working on a project in Huntington's disease. So people may know that Intellia has been working on gene editing for neuropathy due to transthyretin amyloidosis. So they're using a lipid nanoparticle to bring the Cas9 and the guide RNA into a lipid that's coated with ApoE, and then that helps to be taken up by the hepatocytes, goes into the endosome, and then it's broken down, and then the guide RNA and the Cas9 are released and go into the nucleus, and then you get basically what they're doing is knocking out the transthyretin gene to lower the levels. And they worked out in the mouse, and they just published yesterday the paper in the New England Journal of Medicine. So this is hot off the press. And you can see in the patients that they treated, the thyretin level goes to zero, or goes to normal really quickly. And the neuropathy gets better. So it's not just preventing progression, they're actually getting improvement. So this is really interesting. The caveat here is that this is in the liver, and the liver loves to take stuff up. So if there are any liver diseases out there, they're going to be genetic liver diseases, you're going to be able to cure them because this stuff really goes into the liver and works really well. But the other one, which is interesting, which is also another liver disorder note, is urea cycle disorders. And this is being worked on by the group at University of Pennsylvania. that I mentioned. But the amazing thing about this is that they found a kid with the urea cycle disorder, and then within 7 months, they were able to identify the mutation, develop a gene editing program, get it through, as you can see here in the mouse, to show that it was working in a mouse model, then get it into cells to get their editing efficiency up. And then they looked at off-target effects, and they were able to get an IND. We funded this to get compassionate use in the kid, and they treated the kid within 7 months of the birth. So if you think about it, just think about this. You have a disease, you identify the disease, and you treat it within 7 months. Uh, that is profound, I think. And that, I think we should think about this as we go into these genomic therapies. And I'll talk about this, there's not a lot of excuses for slowing down. Now, Terry it will bang on the door and tell you that over and over again, but it's true. We don't have a good excuse for why everybody doesn't have a treatment for their disease, given these tools that are going to be coming out. And this is the process by which the child was treated and doing really well. And I think I showed, yeah. So the ammonia levels basically went down. The kid was treated in the meantime, with protein. There's an enzyme you can give, and there's a protein. You keep the protein out of the diet to keep the ammonia levels up. But you can see before they got dosed, they had some pretty high ammonia level. He had some pretty high ammonia levels, and then afterwards has been able to come off, basically liberalize his protein intake, and doing really well. Now, the big question in these therapies, because everybody that knows what the mutation is, people can build the gene editing process or the new gene replacement therapy in the lab. The big question is delivery. So delivery is the issue for many of these projects moving forward. And you probably heard, I think you heard a little bit today, that there's now technologies where you can screen capsids of the virus. Most people working with AAV may not be the best, but it's out there, so people are sticking with it. But you can screen the capsids to find ones that go where you want. So we funded a couple of groups to develop AAVs that go to the brain and not to the liver. As you may know, there's definitely liver toxicity with the AVs, whether it was the SMA treatment, GAN treatment. So there is something bad that occur in the liver, thought to be in some cases dose related, but not all. But the trick is trying to get more to the brain or to the peripheral nerves and less to the liver. But groups have been able to do this. This is just an example from Ben Dievenman's lab, where they were able to move a capsid with a transferrin receptor binding site to get into the brain. And you can see, it just lights up the brain and the spinal cord really well. So this is working on the capsid. The other thing to work on from the Brain Initiative is we're developing tools to identify enhancers and promoters that are specific for cell types in the nervous system. And you can potentially use those to give you more precise targeting of the cells of interest. So hooking up not to a CMV promoter, but to a promoter that's specific for the neuron that you're interested in. So you get really nice, but these things are dangerous. So one of the groups developed a company that AV capsid that they developed that went to brain and not to liver. The first child who got it died within 4 days of getting the virus, and nobody knew. I don't think anybody knows what happened yet. Nobody has a clue why it happened. I think they did a lot of work in non-human primates, so you would think that if there was anything that you could tell, which would have shown up there. So just to underline that this is still really dangerous territory. Okay, from the gene editing, the great thing about the gene editing program and also the bespoke gene therapy stuff is that all the process of going to the FDA is made public. So these people are making their FDA discussions public, and so the group from Penn who did the urea cycle project I just talked to you about, they've gone to the FDA, and for instance, they've got an approval where for this urea cycle disorder, there's different mutations. They can use the same IND to treat multiple mutations. They're just changing the guide RNA each time, and they're going to allow them to do that. They did try to get ability to do one IND, treat multiple different hepatic disorders, and the FDA, I think they balked at that one, that they'll need a different IND for each disease they go after, but not the different mutations. And then in the prion space, they work with the FDA to get a sense of what they have to do in a non-human primate versus in a humanized mouse. I'm going to end up with this one, and I'm really interested in people's thoughts about this, and that is the idea that should we really start thinking a new way of bringing these therapies to patients? If you can bring a therapy to a patient within 7 months of identifying the disease, is the process we have where you go to a company and, sometimes they sit on it and you're waiting for the phone call. That's not really what you want for the patients. So I've been interested in this paper, which is in Nature, called The Roadmap for Affordable Genetic Medicines from Jennifer Doudna's group. The idea here is that we could really speed things up and keep the costs down if we had a different way of doing things. And what they're proposing is that, and then maybe it's not going to be only for these rare diseases, but for the rare diseases, the IP should go into a nonprofit. And then the nonprofit can work with different types of funders to get the therapy tested and then move that to a space where the treatments can be available at cost. And so if you go to a company, and this company people here, the company has a cost of making the drug, but the company is also trying to make money for the stockholders, and this would be trying to do things just to make the drug and get it out there, and no stockholders involved. So you need some kind of a public benefit corporation or something like that to move the therapy out into patients. This is actually happening in that SCID gene therapy situation where they developed a therapy for this severe immunologic disorder, SCIDs, and it was bought by a company. And the company said, we're not doing it anymore. And for years, it hasn't been available, although it's incredibly effective. But anyway, I just like people to think about that. In the government, I can't do any of this stuff, but you guys are not in the government. And this is kind of from that paper talking about what it really costs to make these things. And it's probably, even if you have some net profit so you can keep your business going, the estimate is about $258,000 per patient, as opposed to $3 million a patient for some of these things now. So anyway, something to think about. We need somebody who has a good finance background to pull this off. All right. Well, thanks a lot. Happy to answer any questions.